Hello friends, today we are going to study about composition, structure and function of carbohydrates part 2. In the last video, we learned about what are monosaccharides, types of monosaccharides, what are epimers, what are stereoisomers, and how we can find the number of possible stereoisomers for a monosaccharide. Now, in this class, we'll study about the configuration and conformation of monosaccharide. First, let's understand one reaction. This is what alcohol, ROH, when reacts with an aldehyde, it forms hemiacetal. Alcohol, when reacts with aldehyde, it forms hemiacetal. Similarly, when alcohol reacts with ketone, it forms hemiketal. Okay, alcohol when reacts with an aldehyde, it forms hemiacetal. Alcohol when reacts with a ketone, it forms hemiketal. Now, similar reaction happens due to which a monosaccharide cyclizes. Now, if you see this structure, this is a structure of D-glucose. What happens is alcohol. So here, alcohol, OH group attached with the fifth carbon. It reacts with an aldehyde, that is first carbon, and form a hemiacetal structure. This structure is known as beta D-glucopyranose. Why pyranose? Because this ring is a pyran ring. Because a glucose straight chain structure, when cyclizes, it takes the basic conformation of a pyran ring. Hence, we can say this is a glucopyranose. This type of projection is known as Howard projection. Clear what is happening here? Formation of a hemiacetal. When the OH group attached with the fifth carbon react with the first carbon functional group, that is an aldehydic group. This is a first carbon functional group. This is the fifth carbon OH group. They react and form a hemiacetal structure. This structure is known as glucopyranose structure because glucose cyclizes over a pyran ring. Similarly, for ketosis, that is fructose. Here again, the OH group attached with the fifth carbon reacts with the functional group which is present at the second carbon, a ketone group. It cyclizes and form fructofuranose. Why? Because this ring structure is a furan ring structure. So it forms fructofuranose. So this how a monosaccharide cyclizes. When an alcohol reacts with an aldehyde, it forms an hemiacetal. When an alcohol reacts with a ketone, it forms hemiketals. And this is the reason, or this is the mechanism behind cyclization of a monosaccharide. Next is, now cyclic sugars have two anomeric form. One is alpha form, another one is a beta form. Now, what is the difference between alpha form and beta form? If you see the structure of alpha form and the beta form, there is a difference in the conformation of OH. In case of alpha form, if the OH attached with the first carbon is on the opposite direction from the OH attached with the sixth carbon, then that anomeric form is a alpha form. If the OH attached with the first carbon is on the same direction to the OH attached with the sixth carbon, that form is known as beta form. So this is the difference between alpha D glucopyranose and beta D glucopyranose. Why gluco? Because it is a glucose structure. Why pyranose? Because it is built on a pyran ring. Why D? Because the OH attached with the fifth carbon is on the right hand side. Why alpha? Because the OH attached with the first carbon is on the opposite direction from the OH attached on the sixth carbon. Clear? Similarly, beta D glucopyranose. Pyranose because built on a pyran ring. Gluco because it is a glucose structure. D because the OH attached with the carbon farthest from the functional group is on the right hand side. Beta because the OH attached with the first carbon is on the same side to the OH attached with the sixth carbon after cyclization. Hence, it is a beta form. So alpha D glucopyranose and beta D glucopyranose. These are the two anomeric form of a glucose. Similarly, it works for all other monosaccharide sugar. Now these two forms, these are interconvertible. That means if we dissolve glucose in an aqueous solution, then at equilibrium, the D-glucose will be in all the three forms, in which 63.6% will be the beta form and 36.4% will be the alpha form, and in very minute we will find a linear form. So whenever we will dissolve D-glucose in an aqueous solution, at equilibrium we will find all the three forms, but we will find beta form at 63.6%, we'll find alpha form at 36.4%, and we'll find linear form in a very minute concentration. This interconvertible nature is known as mutarotation. This property is known as mutarotation. Now next is a chair conformation of sugar. This is an example of beta D glucopyranose, the chair conformation. Now we can see there are two chair conformation. This one is one and this is the second chair conformation. Among two, one chair conformation is a dominant chair conformation. How we will find which chair conformation is a dominant one, or for a sugar, which chair conformation is a dominant chair conformation, in which form the sugar will be available. For that, the rule is the bulky groups. Bulky groups means OH, then CH2OH, which are bigger groups. These bulky groups should be placed or should positioned at equatorial position. There are two positions. One is equatorial and another one is axial. So if the bulky groups are present at equatorial position, that chair conformation will be the dominant one. Now how we will find that bulky groups are present at axial position or equatorial position? Take this chair conformation. This is the axis. So whatever going parallel to the axis is the axial position. This is what? Axis. So whatever going parallel to the axis are the axial position. So these red ones are the axial position. Now in this chair conformation, what are present in this red one? Here H, here also H, here also H, here also H, here also H. So all the non-bulky groups are present in axial position. What are equatorial positions? This is equatorial, this is equatorial, this position will be equatorial, this is equatorial, and this also equatorial. What groups are present at the equatorial position? OH, OH, CH2OH, OH, again OH. So in this chair conformation, all the bulky groups, that is OH and CH2OH, are present in the equatorial position. What is the rule? The bulky group should be present at equatorial position. That chair conformation is the dominant one. So this chair conformation is the dominant chair conformation because here all the bulky groups are present at equatorial position. Whereas if we see this chair conformation, then what we can see? This is non-bulky group, which is in equatorial position. Here also non-bulky group, non-bulky group, non-bulky group, non-bulky group. All the non-bulky group are present at the equatorial position, whereas all the bulky groups are present at the axial position. So this chair conformation is not the dominant chair conformation. The bulky group should be present at the equatorial position. So between both the chair conformation, which one is dominant? The first one is dominant. Because only beta D glucose can have all the 5 non-bulky group in axial position, or in other words, we can say because in beta D glucose, all the bulky groups are at equatorial position. Therefore, glucose is the most abundant monosaccharide in nature. Why glucose is the most abundant monosaccharide in nature? Because the beta D glucopyranose or the beta D glucose is the only glucose where all the bulky groups are found at the equatorial position. Hence, glucose is the most abundant monosaccharide in nature. Next is sugar modifications. Now, this is the first modification. What is happening here? This is a D-glucose. What is the difference between this structure and the second structure? COOH. In glucose, it is a aldehyde, CHO. CHO. Here, it becomes COOH. So, one oxygen extra. So, oxidation. What happened here? Oxidation at first carbon. Oxidation at first carbon. So if oxidation happens at first carbon, it forms acid because it is a carboxylic acid. Sugar converts into acid, and how it converts? Glucose became gluconic acid. So glucose converted into gluconic acid by the oxidation of first carbon. So when it is getting oxidized, that means it is taking oxygen from some other chemical. What is happening with that chemical? That is getting reduced, because this particular chemical is donating oxygen to glucose. So after donating, removal of oxygen is reduction. So this compound is getting reduced. So in this case, what the function of glucose? It is a reducing agent because it is taking oxygen, and because of which the substance which is giving oxygen is getting reduced. So glucose or monosaccharides are the reducing agent. Next modification, oxidation at sixth position, sixth carbon. CH2OH getting converted into COOH. Here also sugar is converting into acid. But how? Glucose is getting converted into glucuronic acid. When the first carbon is getting oxidized, glucose converted into gluconic acid. When the sixth carbon oxidized, glucose converts into glucuronic acid. Ribitol. What is happening here? CHO getting converted into CH2OH, right? More of hydrogen. So it is reduced. If reduction occur, the above two are the example of the oxidation happened in the sugar molecule. Here what has happened with the sugar molecule? It is reduction. Ribitol from ribose. Correct? Xylitol. It is also reduction. From which sugar? You have to find it out. Glycerol. Again reduction. You have to find the corresponding sugar. Myoinositol, again reduction. You have to find the corresponding sugar. So this you have to find. Ribitol, the corresponding sugar is ribose. For xylitol, you have to find. For glycerol, you have to find. And for myoinositol, you have to find the corresponding sugar. You'll get this answer from the carbohydrate video part 1. So in all these cases, what happened is reduction. Next modification, deoxyribose. That means from the second position, oxygen is missing. So deoxyribose. From the ribose sugar, oxygen is missing. Alpha L-fucose, addition of a methyl group. What is happening? Methyl group is getting attached. H3, actually it is reduced only. H3 is getting attached. So formed L-fucose. Next type of modifications are glucosamine, addition of amine group in glucose. Galactosamine, addition of amine group in galactose. N-acetyl-nuraminic acid, addition of N-acetylamine in mannose, manosamine. That means in the structure of mannose. So these are the types of sugar modifications. Sugars are modified, and in this form they are used in various other functions. For example, deoxyribose, you all know where it is used? In DNA. Fucose, where it is used? In blood group. Correct? When we will study the blood group, then we'll study about fucose. So it is used in the blood group as a blood group antigen. And acetylnuraminic acid. It is found on the surface of mammalian cell and function as cell recognition. So all the type of sugar modifications happen inside the cell for its use in different forms. Ribitol, which is a flavin coenzyme component. What else we learned? Glycerol, myoinositol. Both are the component of lipid. These are important lipid components. Xylitol, it acts as a sweetener. So all the type of sugar modifications are done so that these modified sugars are used in our biological system for various functions. Now again, one question from my end for you all. Can you name a qualitative test that determines the presence of a reducing sugar? Monosaccharides are reducing sugar. We perform one test to determine the presence of reducing sugar. You have to name that qualitative test. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.